Who's ready to learn something? Who thought that Friday was the final exam? Okay. It can be. That could be your final exam because the good thing is that there is no final exam. It's just 4 exams. So choose anyone you want to be your final exam. Um, okay, so today we're gonna start learning about signal transduction, and we're gonna do an interactive activity, which should be very entertaining. Because I haven't, like, I think last time I did it, I wrote out what was happening when because I was nervous, and this time I did it, well, we'll see what happens. So, you guys are going to get to stand up and move around, or at least someone will. Anyways. Okay, exams will be back by Friday, hopefully. Don't forget, there are a lot of you. Despite what it looks like in this classroom, there are about 70, I think 74 of you. I thought 75, but I was wrong. And that's a lot of questions to grade. That's why I love doing no multiple choice, but it's completely impossible, right? Just to do all short answer with this many students, unless I had TAs. If I had like 5 TAs, it'd be great. But it's me, I'm the Lone Ranger here. Okay. By Friday, hopefully. If not, I apologize in advance. I know everyone's on pins and needles about how they did. The average on the multiple choice alone was an 80. That's a very good sign. I'm usually happy at a 75. So that means, and I saw as I was scanning through a fair number of hundreds. So that's a very good sign. Because first exams usually are in the low 70s. So I think I have a smart group. Those of you who did not do well, I've already talked to some people who are worried they didn't do well. Don't worry, you can drop an exam. Don't forget you can drop one. The first one is always just about figuring out my style and how to deal with my questions. So you can drop one. But if you did get lower than, say, a 70, come see me and we'll go over the questions because I don't return the questions, but we'll go over them and we'll talk about how you should approach them in the future and how you should change your study habits or whatever it is. Okay. The weekend quiz averages are going up also, which is good. This weekend was about an 80. And I'll go over some of those questions on Wednesday. I forgot to look at the results. Okay. So, here's your generic signaling pathway. It looks like a lot, right? It looks really scary. And there's all these proteins and little fireworks, explosions, things. What's going on here? I just want you to take away some main points with signaling. And once you kind of understand the basic concept, then no matter what pathway I give you, because I will go over many different examples, you'll understand it because it's all the same. It's all something binds to receptor, something makes the signal go into the nucleus and affect gene expression. That's it. So the goal of signaling is pretty much to facilitate cell-cell communication because there's usually some molecule secreted by some other cell or the cell itself that binds to a receptor, and then there's all this magic that happens in the cell, which is usually a protein binding to another protein, and that's mediated by phosphorylation, and that is relaying the message from the outside of the cell, where the signaling molecule binds to the receptor, all the way to the nucleus. So it's inducing something to happen that allows a transcription factor to go into the nucleus, and the end result is to affect gene expression. Okay, so signaling is a way to have a cell respond to its environment by binding to different molecules on the outside of a cell, and that influences gene expression. And this is all very related to what we've talked about already with where proteins go and what they do. So some things I want you to constantly keep in mind, because don't forget, even though we're learning 4 different units, they're not just separate things that happen in isolation. They all work together. So things that you can use that you've already learned, concepts like nuclear import and export, right? Because you need transcription factors to go into a nucleus to affect gene expression. And that's all signaling does. is it does something to allow that transcription factor to go into the nucleus. Endocytosis, because when we talk about receptor downregulation, downmodulation, endocytosis, multivesicular endosomes, and this week's paper is all about regulation of a signaling pathway by sequestering one of the signaling molecules in a multivesicular endosome. So just constantly be trying to integrate what we've already learned. So also signaling is involved in cancer. I wouldn't even say it's involved in cancer. It is like a huge part of the piece of like many different proteins that are mutated in cancer frequently are proteins involved in signaling. So when things go wrong with signaling, sometimes cancer results. Okay, so we're going to go through a few examples just as an introduction to signaling pathways, and we'll revisit them later on in this unit in more detail, but I just want you guys to really get an appreciation that these are different processes that affect gene expression in response to an extracellular signal. And it's usually coming from another cell, but sometimes it's coming from its own cell. Its own self. Okay, so one example is the JAK-STAT pathway. Who loves the JAK-STAT pathway? Good. We need someone to love the JAK-STAT pathway. Without the JAK-STAT pathway, you would have no response to viruses. Okay? Who's had a virus before? Okay. Who's never had a virus? Okay, because you're crazy if you think you've never had a virus. Just making sure. Sometimes we need to wake people up on a Monday morning, ask silly questions, right? Yeah, see, we have someone here struggling. Struggling to stay awake, but he has his coffee, so we're good. I had 2 cups of coffee. You don't want to see me with 3 cups. No. And I'll have to pee a lot, too. Anyways. I just crack myself up so much sometimes. And it's all in here. I'm not even letting it out. Okay. This is what happens when you become a scientist. The crazy increases. Okay, so viral infection activates the JAK-STAT signaling pathway. This is an example of signaling through an enzyme-linked receptor. So we're going to go through a whole bunch of different receptors and different kind of types of signaling, different categories of signaling pathways. And those are what you should be focusing on, understanding the different kinds of receptors and the different types of pathways, not the different individual pathways. If you memorize the JAK-STAT pathway, I'll personally come to your house and slap you around. Okay? Do not do that. But do understand that there is something called an enzyme-linked receptor, and here's an example of that. So you'll have virus infects cells. Cells have immune responses and start secreting signaling molecules called cytokines. Those who have taken immunology or are taking are familiar with that. Those who haven't, who cares, signaling molecule secreted by cells in response to infection. And one of them is called alpha interferon, and it will bind to the receptor. So here's a receptor, and there are these blue boxes bound to the receptor. Those are kinases. And what happens is a signaling molecule will bind to the receptor, and then the kinases that are bound to the receptor, they're not part of the receptor, they're just bound to it. They're going to phosphorylate each other, come together. What is phosphorylation doing? It's activating the kinases, which is then going to phosphorylate the receptor, which creates a binding site where other proteins can come and interact. So phosphorylation is usually all about activating things and creating binding sites for things, right? So it creates binding sites for these STAT proteins, which then get further phosphorylated, and they leave the receptor and they bind to each other, so they dimerize. And then they enter the nucleus and activate gene expression. gene expression. What kind of genes do you think would be induced by the JAK-STAT pathway, just based on what you know about it being involved in the immune response? Just make something up. Maybe some other cytokines, maybe increasing activation of other signaling molecules, right? Increasing expression of other signaling molecules, because then those signaling molecules will be secreted out of this cell to alert other cells that there's a virus infection. Or maybe something involving proliferation, increasing the expansion of cells so they can go further and farther and tell other cells that something's going on. So the whole point is, here we see a process in the cell to hear what another cell is saying, right, because it sees and binds to these signaling molecules secreted by other cells, and affect gene expression appropriately. Do something to increase the expression of genes that will help fight the virus. Okay, so when we think about signaling, don't get bogged down with this binds to this, binds to this, binds to this. Who cares? Who cares? It's all the same. It's all about getting some transcription factor into the nucleus to increase gene expression. Okay, does everyone get that? Okay. So I'm going to talk about, all right, so this is an enzyme-linked receptor. And when we talk about an enzyme, I'm usually referring to a kinase in this context. And a kinase, what's a kinase? It's right up there. It phosphorylates, right? So it's an enzyme that phosphorylates other proteins. And when you phosphorylate another protein, you're affecting a protein's activity or its ability to interact with other proteins. So signaling is all full of kinases phosphorylating, and phosphatases dephosphorylating, and ubiquitin binding to things and degrading things, and it's fun. Okay. So here's a question for you to think about. I want you to talk about it for a minute or two, and then I actually am going to make this an extra credit. And it's an extra credit that I'm allowing you to cheat on by talking about it with your friends for a minute, just to make sure everyone's on the same page. But the reason it's going to be an extra credit is because it's going to require you to do a little thinking and possibly a little bit of research. So I'll go through it in a minute, but to get the extra credit, you need to get it to me by noon tomorrow, and it needs to have a referenced example, a reference to what your answer is. Okay? So, activation of the JAK-STAT pathway results in dimerization of STAT, which then goes into the nucleus to activate gene expression. Think about import of proteins into the nucleus and what you know about it. Okay, you guys all know this because we just learned it and we studied it and we all did our extra credit if we had time. What mechanism regulates whether STAT will be nuclear or cytoplasmic? How does this affect gene expression? And then design an experiment to test your hypothesis. So talk to your neighbor for a minute about this, and then you'll get it to me tomorrow, and we'll go over the answer in class tomorrow. Tomorrow on Wednesday. Okay. Uh-oh. Too much quiet for my liking. Okay, guys, so it's a lot quieter in here than normal, which makes me think that some people, either people are just like, you know, it's an extra credit, I'll deal with it later, or they're like, I have no idea how to even think about this. Who's of the first who's like, I'm just going to think about it later? Well, it'll be posted. Who's like, I don't even know how to think about this? Okay. Okay, good. I have one hand up. Good. Thank you. Good. But you're not sure. Okay. So, who wants to tell me what what direction they think they should go in thinking about this? Who wants to just to help their classmates get extra credit? Yes.
Is there any involvement of it having an NLS?
Probably so that's exactly where I want you to be going, right? So think about imported proteins into the nucleus. So is there any involvement of it having an NLS? That is definitely the direction you should be going in. And it must have an NLS. But now think about the next question. What regulates whether STAT will be nuclear or cytoplasmic? Okay, yes?
What do I do with RAN gap and whether it's activating the GTP to import?
Okay, so I just had a comment about RAN gap and RAN GAF and whether import is going to happen or not. I want you to back up from there and think about where RAN gap and RAN GAF are in the whole process of import, and think about what's going to determine if the protein's ever going to be imported. Because RAN gap and RAN GAF regulates the general import and export of proteins, but this is specifically STAT, dimerized STAT. So STAT by itself lives in the cytoplasm normally. And let's go back. And with this whole pathway, what ends up happening is you get phosphorylation of things and dimerization, and it's only that phosphorylated dimerized STAT that goes into the nucleus to activate gene expression. So what you need to be thinking about is, where is the NLS? What is the NLS doing? Why is it not? Why is STAT not always? If it's just that there's an NLS in the protein, why is STAT not always in the nucleus activating gene expression? Why is there some kind of regulation that's based on phosphorylation and dimerization? Okay, and I'm sure it's been studied. I'm sure there's plenty of papers on it. And so the question is, this regulation on of STAT. How does this affect gene expression? Okay, so if it's nuclear, it's going to have one effect on gene expression, and if it's cytoplasmic, it's going to have a completely different effect on gene expression, right? And then you have to design an experiment, which really goes right down to what we always talk about how to test if there's some aspect of a protein that you think does something. What do you do? So does that clarify things to those who are confused? Okay. So I'm really trying to get you guys to keep integrating the information that you're gaining in this class with new concepts that are coming up. Email me if you're still completely confused, but I think most people in this room seem to have an idea of where to go. Okay, here's another pathway. And this is an example of a pathway that's dependent on regulated proteolysis. So what is that? What does regulated proteolysis mean? Yes. Breaking down proteins in a regulated fashion. So before I even go through this, what kind of protein do you think is sometime, well, it doesn't matter. Let's go back. That's probably way too much to ask right now. Okay. So, I covered something up. Okay, under here it says something else. NF-κB pathway is involved in regulation of gene expression involved in the immune response. So more immune response related stuff. And with gene expression, I think immune response is the easiest way to understand the alteration of gene expression, because clearly you don't want an immune response when you don't have any reason to have one, right? So that's all, that kind of makes it easy to understand your environment regulating gene expression. So in NF-κB pathway, you have some kind of ligand that binds to some kind of receptor, and then there's a whole bunch of proteins that assemble. So you've got this scaffold protein, which a scaffold protein is a large protein that a bunch of other proteins assemble on to do have some function. And then what ends up happening is you've got this IKK kinase that becomes activated. And then the activated kinase phosphorylates this other complex, which activates it, which then phosphorylates this other protein called IKB, which is the inhibitor of kappa B. So it's an inhibitor that's phosphorylated. And when the inhibitor is phosphorylated, it's ubiquitinated and degraded. And that allows the NF-kappa B transcription factor to enter the nucleus and activate gene expression. Do you see how this is very similar to the other one I just showed you, right? Where you have something, you've got something binding to a receptor, you've got a bunch of phosphorylation, and then something goes into the nucleus to activate gene expression. The difference is, in one case, you have phosphorylation and dimerization, and that dimer goes in. And in the other case, you have phosphorylation and removal of an inhibitor, which allows a transcription factor to go in. Okay, so this is an example of regulated proteolysis. What are we regulating? What the proteolysis of what here? An inhibitor, right? Right. So when that inhibitor is not being degraded, it's inhibiting gene expression. So when you have interaction with the receptor, you have this whole signaling cascade that results in degradation of the inhibitor, allowing the transcription factor to activate gene expression. So what is a likely role of I-kappa B, which is the inhibitor, I is for inhibitor of kappa B, in preventing import of NF-kB, which is the transcription factor, into the nucleus? What do you think I-kappa B normally does? How does it inhibit? Don't forget, in one case, NF-kappa B is cytoplasmic when the inhibitor is there, right? When you trash the inhibitor, it goes into the nucleus, yes?
Well, a common way of inhibiting genes is having an inhibitor bind to a piece of the DNA so the RNA polymer can't cycle through it.
Okay, so definitely once you're in the nucleus, you do have things where things are binding to genes and preventing gene expression. But in this case, the signaling pathway, in terms of the inhibitor I-kappa B and NF-kappa B, that's all cytoplasmic. And so you're absolutely right in general mechanisms of inhibition of gene expression. But what about even getting that transcription factor into the nucleus? Yes?
It's just covering up the import signal.
Right, really. That's really all it's doing. The inhibitor of NF-kappa B binds to and covers up the NLS. Right? So if there's no NLS, it's cytoplasmic. When you degrade the protein that covers up the NLS, it becomes nuclear. It's such a simple mechanism, right? Who's like, what is she talking about? Is anyone sitting here going, why am I even taking this class? You know what? I promised I would take this poll. Let's take a break from talking about this for just 1 second. I did this in the beginning of Mole Bio, and I forgot to do it in this class. How many people here want to go to graduate school, like to get a PhD to do research or master's? One. Oh, big handful. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Okay. Probably a good chunk of the class. How many people want to go to professional school? Med school, vet school, pharmacy, physical therapy, more of the class. I'd say probably about 20. Really? Aren't you so sad that you sit in front? I always pick you out. Where's the other one? Okay, you're quiet and in the back. Too much. Okay. Sorry, I just like to pick on people. How many people have no idea what they're going to do with this degree? Everyone has a plan? I'm so glad you guys are so organized. Okay. How many people are really regretting? Oh, how many people are secondary ed? Very good. Very good. These are always some of my best students, my secondary ed students, because they know how to break down, which is important if you're going to be teaching students. And so how many people are wondering why they're a biology major and wish they could not be one, but feel stuck because they're about to graduate? Really? Okay, some smarty pantsers are raising their hands. Okay. I got many more hands in 409 on that one. I think I had at least 15 or so who were like, why? Okay. Don't forget, graduate school is a way to change your direction. Anyways, okay, let's move on. I did want to know how many people are just hating being here, but it looks like it's only a few people. Well, let me take that back. You could all hate being here. Okay, thank you for that quick demographics there. So this is an example of a pathway dependent on regulated proteolysis, because what's being degraded is the inhibitor. And this is very similar to Wnt signaling in beta-catenin, which is the paper we're reading in recitation. So as we read this paper this week and as we discuss it in class, I want you to think about what's being degraded and how is that controlling gene expression, or what's not being degraded, how is that controlling gene expression. Okay. Mutations in signaling pathways frequently occur in cancer. And so one example is the Wnt signaling pathway. There's so many. God, I can go on NF-kappa B. Everything, probably every signaling pathway, there's some mutant somewhere along the way that's associated with some kind of cancer. And so an example in the Wnt signaling pathway is this APC protein right here, which is part of this destruction complex. So let me go through it real fast, right? So we have Wnt signaling, there's here are your receptors. There's actually 2 receptors that work together, and when Wnt, which is the extracellular signaling molecule, comes along and binds, then you have activation of this other protein, which then results in this whole complex being turned off, and that's what we're reading about in the paper this week. This whole complex being turned off because GSK3B needs to be inactivated. And then that allows beta-catenin to go into the nucleus, because without Wnt, what happens is this complex is active and you end up degrading beta-catenin. Okay? When you don't have, and beta-catenin is your transcription factor. When you don't have beta-catenin, then it can't go into, if you don't have a protein, can it go into the nucleus? Right. If a protein's been degraded, will its NLS bring it into the nucleus? Okay. Okay. Who doesn't know what that question means? Okay, good. Right. So does everyone understand how important ubiquitin is now? So you have, again, degradation of the transcription. Here you have degradation of the transcription factor itself, not an inhibitor. It's different from NF-kappa B. You still have degradation of something. Okay, so in colon cancer, 80% of colon cancers have APC mutated. Do not confuse this APC with the APC that you'll learn about later on in the semester. This is just a specific protein that's involved in the destruction. It's called the destruction complex. Do you like that? Destruction complex, which is involved in degrading beta-catenin. So when this is mutated, beta-catenin is not degraded. If beta-catenin is not degraded, it can do what? Does it activate or inhibit gene expression? I heard someone say activate. Yeah, so if beta-catenin is not degraded, its NLS is out there in the open. It goes into the nucleus, and it binds to it's basically a co-activator that interacts with the transcription factor that's there, and it displaces a repressor. And it activates gene expression of tons of different genes, one of them being MYC, which some of you may have heard of before, but either way, lots of genes associated with cell growth, actually. And cell growth, when it's not regulated, results in what? Cancer, right. Just answer my question. What is the outcome of its effective APC? Just think about it, because you guys are going to read the paper. You're going to understand a lot more about this signaling pathway this week. And so when you're studying for this exam, come back and think about this question. Any questions so far? Good. Okay, so another thing to drive the point home. Unregulated signaling can result in cancer. Another example is chronic myelogenous leukemia, and that's related to 2 genes, a translocation which results in 2 genes, BCR and ABL, being fused together, and it ends up with this fusion gene, which results in basically unregulated tyrosine kinase signaling activity, which affects many different proteins. Because it's not a receptor, it's not a transcription factor, it's actually something in the middle of the pathway, and it affects a whole bunch of signaling proteins involved in cell growth, differentiation, apoptosis, all the things that you need to be regulated pretty tightly so you don't have cancer, right? Cell growth, differentiation, apoptosis, right? So they developed actually a drug. So this is one of your better leukemias to get, like if you had to choose. leukemia. I would choose this one because they developed Gleevec. Who's had leukemia? Good, thank you. It's good. You know, you don't want. I'd hate it if all you raised your hands. So I'd say something bad about Towson, Baltimore. But actually, I knew somebody who did have leukemia, and they had this kind, and they took Gleevec, and I mean, it's a good one. It's a good one. So the way it works is you've got BCR-ABL, which is this protein that came from this translocation, and what it does is it's a kinase, and it phosphorylates some substrate protein, and when that is activated, then you have this signaling for cell proliferation and survival, lots of cells, leukemia. But with Gleevec, what it actually does is it binds the ATP binding site and prevents activation of the kinase and phosphorylation of the signaling protein, and you'll have no signaling, so you don't have all that cell growth and the survival. So pretty awesome. Yeah?
Just a curiosity question. What is it for ALL? That's what my brother had.
I don't know.
I don't know.
No, but I can look it up. If you email me, why don't you look it up?
Okay.
ALL, adult, what is it?
Acute lymphocytic leukemia.
I think my friend had that. Did he have a bone marrow transplant?
Mhm.
From himself?
From
Donated?
Yeah.
My friend had, I think she had that and had an autologous from herself. It's really interesting, yeah. 16th chromosome? Oh, and do you know what gene is affected? Oh, we should look that up. We should do a leukemia project, see what genes are affected. Unfortunately, so this would have been a much better leukemia probably. But let's look up what gene. So you said 16th chromosome, there's an inversion. So let's see what everyone write it down because it's going to be extra credit. Oh my God, 2 extra credits in one day. I feel bad after exams because people come to me crying. So, inversion involved in ALL, AML, and then you had ALL.
My brother.
Yeah, not you, your brother. Sorry, it was you. You mentioned it. And so, what mutations are involved in those leukemias, and what signaling proteins? I'm guessing there's a signaling protein that suddenly is not acting the right way, and that's what results in the leukemia. And so, people send me what proteins are messed up, what's mutated, and how does this affect signaling? Okay. Yes.
Are these separate or together?
That's one question. Yeah, both of them. Because I think it is really, cancer is so interesting to me because it's usually one little point mutation that results in overactivity or underactivity of some kinase or phosphatase, and that's so if it's in the middle of a signaling pathway, it's really bad news. Okay. AML and ALL, AML and ALL. I know there's a lot of who knew how many leukemias there were. There's a lot of leukemias. Okay, so we're gonna do a quick activity. On that note, okay, so I need volunteers to come up here. Let's see. Who wants to get up and move around, or who wants to stay seated? Who's feeling like they want to move? Come on. I need someone to be. Okay, where's my receptor? I need someone to be my receptor. Come on. Okay, so receptor, stand over here. It's a perfect place. Okay, so who's going to be my extracellular signaling molecule? Come on up. We got to do fun things, otherwise we fall asleep, right? Otherwise, I'm going to have to drink a third cup of coffee. Okay. So after that, we're going to have, that's not a complete, they're missing some phosphates. Who wants to be a scaffold protein? Okay, come on. Okay, we're going to do it as we go, kind of. So what's going to end up happening is we're going to have signaling molecule binding to receptor, and scaffold protein is then going to bind You know what we can do? Put it up on the board with our magnet. So put your receptor up top and your signaling molecule, bind it to it, and then you can sit down and you don't have to all stand up. And then put your scaffold protein, kind of bind it over here. There we go. And it's gonna thank you. And okay, so we have activation of our signaling pathway. Can I have some other volunteers who want to be relay proteins of some sort? Okay, one more. One more. Thank you. You can be relay too. It's like thing 1 and thing 2. Who's going to be thing 1? Anyone? That's like the most cliche. Come on up. Halloween costume for twins. You know that? Thing 1 and thing 2. Okay, so these are just other proteins. So you're going to have phosphorylation at some point, and I didn't make enough phosphate, so forget that. So you have phosphorylation. That's going to allow this scaffold to come, right? And then it's going to recruit relay 1 and relay 2. That's going to bind to the scaffold. So the whole point of the scaffold is to bring other proteins in. Thank you, guys. And then you're going to have, oh, right. And so somewhere along the way, like a kinase is going to be activated. Who wants to be my kinase? Anyone? We're doing generic signaling 101. Uh-oh. Everyone who hasn't come up fails. All who came up will get an A. You gonna make me do the song and dance by myself? Who's scared of standing up?
Can you label those? I can't read that writing.
You can't read my writing?
It's like too small.
Don't offend the teacher. Extracellular signaling molecule. Receptor. Okay, extracellular signaling molecule binds to receptor, which results in phosphorylation of receptor, recruitment of some scaffold protein, super generic, right? Scaffold protein, which then recruits 2 proteins. I'm calling them relay 1 and relay 2 because they're going to relay the signal. Relay 1 and relay 2. And then they're going to recruit some kind of kinase. Okay, so since everyone is lame. Yeah, come on up. A little peer pressure. I mean, I guess I'm not your peer, so a little teacher pressure. So let's recruit the kinase to our scaffold protein. Let's put it right here. Okay, thank you. And what else do we have? Then we're going to have, that's going to end up phosphorylating this protein that's just going to transduce and amplify, which means it's going to bind to other proteins and phosphorylate them. Who wants to transduce and amplify? God, you are lucky I don't make you get up and do yoga. Okay. Transduce and amplify, and then that's going to send the signal to others. In every single case, you're really having, there's some other phosphorylation going on here, which is recruiting this, and this is phosphorylating that, and then this is going to phosphorylate that and that. And then, what's next? Not that. Okay, then we have this situation here. That's an inhibitor bound to a latent gene regulatory protein because that's just the pathway I chose. And so what's going to happen is then there'll be another kinase. We're going to steal our kinase and go put it over here. That's going to then phosphorylate our inhibitor, right? So this will activate this. So these two, there are ways to spread out your signaling to different pathways. So maybe this one will activate this, and this will go to another pathway. So we forget about that one. So then we have activation of some kinase, right? Another phosphorylation, and then we have phosphorylation of inhibitor. Okay, so if we phosphorylate our inhibitor, what's going to happen? The same former students to this. Oh man. There you go, polyubiquitin. And then what happens to that? It goes to the proteasome. And then latent gene regulatory protein. Oh, right. Signal to other pathways. Sorry, guys. This was supposed to go down here. Signal to other pathway. Okay. Latent gene regulatory protein is liberated. And it can actually be, it goes into the nucleus. So let's make our nuclear envelope with our nuclear pore. Remember we learned about this? And it can go into the nucleus where it'll either do something which will result in, here's your DNA. I guess it should be helical. Does that look helical? And it will bind to the DNA. And what will it do? It will recruit in some way. What it'll do? Bind to DNA, recruit RNA polymerase. And then you get gene expression. Okay, transcription of whatever gene. Now, one thing I keep, thank you to everyone who participated in the chaos. I appreciate putting yourself out there is hard. Who loves to do karaoke? Okay. Now, why you didn't come up, I don't know. If you love karaoke, who likes to do karaoke when they haven't had a drink? Oh, well, some of you guys came up anyways. Okay. So the one point, though, I want to make about all of this is, and those of you who've taken 409 or are taking 409 right now will understand this a little more. You've got this latent gene regulatory protein, which pretty much is a transcription factor. So this can actually be the same protein. And it binds to the DNA. I keep saying bind to the DNA. What is it binding to in the DNA? Does it bind anywhere on the DNA, or does it bind to a specific place on the DNA? Right, it binds to a specific place on the DNA, and that specific place is unique to a certain gene that needs to be expressed. And that's how signaling results in expression of certain genes in response to the environment. For example, with NF-kappa B involved in expression of TNF-alpha, or which some kind of gene involved in the immune response. Because there are sites on the DNA that transcription factor binds to, and they're in the promoter region, and they bring in RNA polymerase, or they kick off in some sort of inhibitor, or they recruit more activators, or something like that, that allows gene expression of a certain gene. Right? What would happen if it just bound to anywhere on the DNA? Nothing helpful, right? It has to be in a certain place. Okay, there's our activity for the day. I'll get that later. Okay, let's move on. Okay, so cells communicate through binding of receptors, through the binding of receptors by signaling molecules. That sounds so bad. Cells communicate through signaling molecules binding to receptors. And those molecules are usually secreted by other cells. Sometimes it's secreted by its own self. And then there are two kinds of signaling molecules, okay? There are hydrophilic signaling molecules, also thought of as ligands, right? And they bind to cell surface receptors. And then we have signaling that looks like this. Kind of. I really love my sparkly ubiquitins. My former students, wonderful. And then there are these small hydrophobic signaling molecules that actually diffuse across the plasma membrane, so there's no receptor, and they bind to intracellular receptors, which are often, nuclear receptors. And we'll learn about that a little bit. And those are usually things like hormones. Hormones work that way. But in either way, what's happening in the end? Something's going into the nucleus, something's binding to DNA at a certain site, and activating expression of a specific gene, because all of this came from outside the cell, right? The message started outside the cell. Any questions about this? About everything we've talked about? I feel like I just keep talking. That's my job, but, you know. Okay. So signaling molecules can remain bound to the signaling cell, or they can be secreted and they can act over long or short distances. You can have this contact-dependent signaling, which is similar to what you see in the immune response. Remember, we talked about CD4 downregulation by HIV, NF, and all that. Remember CD4, it was a receptor that's involved in the immune response. Well, that's this contact-dependent signaling because you've got a cell with a receptor that binds to another cell that has a different receptor, and that interaction activates this whole signaling cascade. So it's not always a secreted molecule. You can have paracrine signaling, where you have a cell that secretes some kind of protein that that binds to receptors on cells in the area. So it's a local mediator. You can have synaptic signaling, which I don't know anything about because I don't know neuroscience, but it has to do with neurotransmitters, synaptic signaling. And you can have endocrine signaling, which is when you have basically hormones secreted and they travel over long distance, usually in the bloodstream, over long distances. Quite different from paracrine signaling, where it's in the local area. So secretion of a hormone goes to the bloodstream and binds to receptors on target cells. So different cells, they respond to combinations of signals, and that directs the behavior of the cell. I'm really trying to drive a point home. You guys hopefully all got it. Right? So in some cases, you might get signaling that will promote survival. Sometimes you'll get signaling that will tell the cell to divide. Right? So signaling that promotes survival will activate genes involved in what kind of process? Or I should say not activate, inhibit genes involved in what process? What's the opposite of survival? Death. What process is involved in regulated death? Apoptosis, right? Right? Sometimes you have to think backwards and upside down, but you get there, right? So some signaling will promote inhibition of genes involved in apoptosis, which will result in survival. That kind of signaling sometimes is also dysregulated in cancer, right? Because you'll have all this survival. Sometimes signaling will, different molecules will bind to different receptors, and you'll get signaling that will result in cell division, right? And we see that a lot in the immune response. This might be signaling that activates gene expression involved with what? What needs to happen before cell division? Well, that's really right before cell division. Okay, cell growth, cell division. Before mitosis is what? What do you have to copy before mitosis? We didn't learn DNA. So we didn't learn this yet, but you've learned this before, right? You know, S phase is before M phase. So usually it will stimulate gene expression of things that are involved in copying your DNA, and then all the things that need to be set up before mitosis. Some different signals will involve in differentiation, right? So one cell, maybe a stem cell, becoming some other kind of cell, obviously. And then some, look at this, lack of signaling. Sometimes a lack of signaling will result in apoptosis, and also some signaling will result in apoptosis. And that's the picture. Okay. I think I keep getting told that when time's up is actually 2 or 3 more minutes left, so we're gonna go for the gold here. So different cell types, they respond differently to the same signals. So you can have a heart muscle cell and a salivary gland cell and a skeletal muscle cell, and they'll all be bound by acetylcholine. And the heart muscle will decrease contraction, and the salivary gland protein will secrete. What does salivary gland cells secrete?
Saliva.
Oh, good. Saliva. And skeletal muscle cells will contract. And the whole point is the same signaling molecule, but they're different cells and have different effects on different cells. Okay, I guess we'll finish this on Wednesday.